Welcome everyone to this webinar hosted by Anybody Technology. And the topic of today's webinar is about integrating predicted ground reaction forces in IMU systems for kinematic analysis. My name is Christopher Iversen and I work here at Anybody Technology. Today I'll be the host of this webinar and I'll be assisted by my colleague Hamed Shayestepur during the Q&A session in the end. So in the Anybody webcast series, we usually host presenters who have worked with the Anybody modeling system to cover different use cases. We do this to help everybody out there who are using the system or who are just interested in doing something similar or catching up with people who are using the software. And in today's webcast, I would like to introduce our speaker, Hugo Giambini, who is an assistant professor and director of the Musculoskeletal and Orthopedic Biomechanics Lab at the University of Texas at San Antonio. And Hugo will provide us with insights on using musculoskeletal models for fracture risk prediction by integrating predicted ground reaction forces and IMU systems. And this presentation will start in a few minutes or so. But just before we start, I will give you a general introduction and overview of the Anybody modeling system, if there is any of you who are unfamiliar with the software or musculoskeletal modeling and simulations in general. So first of all, I would like to draw your attention to the control panel, which appears on the right side of your screen. And you are very welcome to submit questions and comments via the question panel during the presentations. And we will try to address these questions at the very end of the webinar. And in case we do not get to answer all of your questions, we will try to do so by email within a reasonable time. And I already agreed with Hugo that I'll send all the questions to him afterwards. So if there's anything we don't get to answer, then he will send you an email directly. So let's begin with having a look at what the Anybody modeling system actually is. Well, the Anybody modeling system is a software that allows you to do musculoskeletal modeling and simulations. And as input, it takes motion data as kinematics and forces, and it calculates the internal body loads as joint reaction forces, muscle activity, and metabolic energy. And at the moment, Anybody is really used in a wide variety of areas and applications. And here is a few examples of this. It could be movement analysis, product optimization design, the field of sports optimization, orthopedics and rehabilitation, ergonomics, which could be with and without some sort of assistive device, as for example an exoskeleton, and it could also be in the automotive industry. So we really have clients all around different industries. And just to give you an idea about the typical workflow in Anybody, I'll just let this one run to the end. So basically you provide the recorded motion data as input, and then you can use the body models which you or others have built, and then you can provide some kind of environment, which could be a special type of equipment, or for example an exoskeleton as it is in this case. Then you can use Anybody to combine these things and solve the muscle recruitment and run the inverse dynamic simulation. And this basically means that we go from motion to calculating the internal body loads and the interaction with the environment in some cases. You can then output the result and use it for some kind of post-processing with, for example, it could be with some kind of finite element tool. But there's also many users who just close this loop completely by doing some kind of design optimization and then run this cycle multiple times. And this brings me to the end of the introduction, and I'll hand over the word and presenter role to Hugo instead.
Thank you for allowing me to present some of the work that we've done over the past year or so, and thank you all the attendees that were able to join us to see what we've been working on recently. So as Christopher mentioned at the beginning, we will be talking about MSK modeling, but mostly how the predicted reaction forces and kinematics can be used in this modeling or in modeling software to get outputs that could eventually be used for fracture risk prediction. So, excuse me. The work that I will be presented is or was mainly performed by my grad student, who's now moved on to a postdoctoral position at another university. But the interest in our lab relates to vertebral fractures. And here you can see a schematic of a skeleton with trabecular bone, which is the bone, the spongy bone inside our skeleton. And as we age, there are many factors that lead to fractures. You can see that in the video, our spine changes. We become a little bit more kyphotic with age as we get older, and this affects the loading in the spine. So structurally, now the spine is different. As we age, we also have a reduced bone integrity. So what our trabecular bone becomes is lost as we age. Our cortical bone becomes thinner. Our muscle also weakens. So there's this cycle that affects the loading in the spine. And if the loading in the spine is affected, considering that now the structural bone is weaker, well, all of these might eventually lead to an increase in fracture risk or increase in fractures. And this is specifically relevant in the cases of osteoporosis, which is an age-related systemic skeletal disease, which you can see here that we have two vertebrae, schematic vertebra, one's a normal, the other one's an osteoporotic vertebra. And you can see that there's low bone mass, which eventually leads to decreased bone strength. There is architectural deterioration. So in addition to loss, we get thinning of trabecular, thinning of the cortex, depending on the bone location, femur, vertebra, etc. And again, all of these leads to a significant increase in fracture risk. And in the US, it is estimated that by 2030, the prevalence of osteoporosis and osteopenia will rise to 13 and 57 million people. So it's a significant issue that will affect the quality of life of everyone. It will affect costs. So there's something that in our lab we're very eager to work on in terms of prevention. So not treatment. We are not interested in treating vertebra. We're interested in how can we eventually design a process or a workflow that will allow us to predict who's going to fracture. Now, if you, to get you a little bit more familiar with how the process is in the clinic, when you go to the clinic now, we use DEXA, which is dual X-ray absorptiometry, as the gold standard for fracture risk or osteoporosis diagnosis. And if you look at this, this is just a 2D X-ray image of the spine. We only get bone mineral density estimations of the lumbar 1, lumbar 2, lumbar 3, and lumbar 4 regions. So it's a 2D picture, which has a good correlation, about 50, 65%, let's say, with structural failure loads of the bones. But you can see that there's a lot of limitations. Some of these limitations include that there are cases where you might have a degenerative spine, and with degeneration, the bone density increases in specific regions of the vertebra. So if you look at this vertebra that has some degeneration, so for example, if you look at the L1 here at the top right corner, you see a very whitish region. Now, that might be degenerated bone or an osteophyte, and that will increase the bone mineral density that is seen on that vertebra, but that's an artificial increase in BMD. So having a high BMD doesn't necessarily mean that you're going to have a lower risk for fracture, because this is just a degenerated region of the bone where the rest of the bone might be weaker. So there's an overestimation of BMD in the lumbar spine. Then the other limitation that it has is that it's technician dependent. If you look at this horizontal lines in the X-ray, that's placed by technicians. And we're humans, and we can place the line at different angles, at different heights, and all of that is going to affect the BMD that we get. So there's a human error associated with the process. Then I don't think that I need to emphasize that having it be 2D also leads to limitations that we can potentially overcome with 3D measurements and, of course, MSK modeling. And to restate, there's a 40 to 65% prediction of failure loads using DEXA, but I think that we can do more. And we can do more not only in the sense of increasing this predictability of failure loads, but also to see what are the loads in the spine based on different activities, right? Because based on the different activities, how our spine is curved, as I showed in the first slide, all of these aspects will affect the loads at the different vertebral levels. And depending on the structural bearing capacity of that vertebra and the load that we impose, we may have a fracture risk associated with different regions of the spine. So this is what we're trying to overcome in our lab. How can we improve on this process? So the first question that we ask ourselves to try and move our workflow forward is, what is the risk of fracture based on different activities of daily living? And the first step to address that question was that we wanted to estimate spine joint contact forces from these ADLs. And this, again, was what I will show today is mainly work that was published in Journal of Applied Biomechanics. It's the work primarily by Maria Prado. She was a graduate student in our lab, and she moved on. But most of the work that I will show, or all of the work that I will show in this webinar presentation using MSK modeling arises from this paper. So why is MSK modeling important? Well, to estimate spine loads is very difficult in vivo, as we all are aware, all of us that are signed up in this webinar. We know that this is a very difficult process. And what I want to show is this person that you see in this figure. And you can see on the circle, on the red circle, there's a kind of a black horizontal marker. So that's a pressure transducer. So this person, this was in 2001. It was a study by Wilke et al. And this person was, I would say, brave enough to allow for a pressure sensor to be placed in the intervertebral discs. And what the study did is it measured pressure or intervertebral pressure with different motions or daily activities that we would perform daily: flexion, extension, lateral bending, etc. You can see that this is a study that's almost 25 years old. I would imagine that to get subjects for this type of study is very difficult, because to have a pressure sensor placed in the disc is not something that I would at least look forward to. But this was a study that provided a lot of information about the loads in the spine. But what is something that we have today that allows us to not go through this process? Well, we have MSK modeling, right? But how does MSK modeling work? Well, MSK modeling needs some input, right? And in this case, the input is mostly based on motion capture. And the motion capture that's routinely used for this type of processes is based on reflective markers. So you see this subject is in this laboratory setting. So it's a closed setting, maybe a clinic or an industry or a lab at a university. But it's a closed setting that has a lot of cameras around the subject. And what the cameras do is they measure where the markers that are placed in this subject are in the environment. And that's how they estimate eventually the kinematics. And those kinematics then are used as input into our modeling. But what we wanted to do is, because we want to move outside of the lab, we want to see, well, are there, and if you're familiar now, there are a lot of markerless approaches. But in our case, we wanted to look at IMU systems, or inertial measurement unit systems. And we specifically use a tool and a software, it's called Xsense, that it allows us to omit the use of cameras. And the idea is that we don't need to be in a lab, in a closed environment. We can be outside. And also, importantly, we don't need force plates. If you see this person is walking on this black mat, let's say, and we can assume that those are force plates. But by implementing IMUs and this template from antibody, we can predict this reaction forces. So that allows us to move forward into our fracture risk workflow process, that we now don't have a need to do everything in-house, or by in-house, I mean in our lab, have the subject come into our lab. We can go outside, do it in, I'm gonna go out on a limb here with parks, or even the clinic, in the hallway. And that optimizes the workflow, because to have subjects eventually come to the lab, that adds time. If we look at fracture risk, we're talking about elderly subjects, so we want to avoid having to have them come many visits. So if we can move this tool to the clinic, where they go have their routine clinical visit, okay, now let's set you up with IMUs, let's have you do some activities. That adds to improvement in the workflow process. So what we did is we collected data from 11 healthy subjects to try and look at our process. But the first thing we did is we implemented what's called a spinal mouse. And the idea of using this spinal mouse is we wanted to look at subjects that had a normal curvature, so that I don't know if the videos I'm seeing it, that it's cutting. I don't know how clear it's seen. But the idea with this spinal mouse is that we can roll the spinal mouse in the back of the spine and get their curvature. And by looking at healthy subjects, then we see that, well, now at least our population that we are studying is normal. And eventually we can implement the same tool and technique on pathological subjects to look at their kyphosis on lordotic changes. So when we look at the spine of these subjects, we quantified kyphosis, lordosis, circular inclination angles, and we found that everything was within normal ranges. So now that we know that our inclusion criteria only, population that's included in the study includes only healthy subjects, then we went ahead and captured kinematics with both of our systems. And what we did is we set up the subjects with markers, so that we brought them into the lab, set them up with markers, as you would routinely do in a kinematic assessment. And we also concomitantly set them up with this Xsense system, or IMU system. You can see the subject has this orange squares in the back. And in this schematics, you see that all the green relates to where the markers are placed when we use the camera system, and the orange relates to the IMU-based system. But the nice thing about the IMU system is that they're vests or ties or bracelets. So if you look at the left arm in this subject, you see a black bracelet, and you can hopefully see a orange marker. So there's specific locations where we just strap this. It's very easy to use. And the idea here was to compare, well, how does IMU kinematics compared with what we see as a gold standard, which is a marker-based system. And eventually what we did once we obtained the kinematics is we used them as input in our MSK modeling. And what we had the subjects do is first some static positions. So we had them stand, hold arms at chest with weights, extend their arms with weights. So these were all static positions. And then we had them do some dynamic motion, flexion, extension, lateral bending, rotation, walking with weights. And from these, you can see here some a sample of our modeling approach using this relates the marker-based process. But from this, what we wanted to get out of was a comparison between the kinematic output from antibody based on the two systems, the marker-based and the IMUs, the contact forces on the L4, L5 from antibody using both systems, and see how the ground reaction forces compare. Because remember, the marker-based, we use force plates. On the IMU system, we use predictions of these reaction forces. So we wanted to see, well, how good were the predictions of ground reaction forces, which will eventually have a role in the whole dynamics process and estimation of forces in the modeling. So the first thing we did is, based on the participant from the Wilke study that was 72 kilograms and 174 centimeters height, we tried to recruit one of these subjects that matched that weight and height, because we wanted to see, well, how good is the modeling in actually predicting in vivo experimental forces. And what you see here is the in vivo data in black, the marker-based and the IMU system. And you see that there's a fairly good match between our forces, regardless of whether we use marker-based or IMU-based. So we were very confident that the modeling approach was working well, because if we can always get data out of the model. Now, how reliable, how accurate is that data? Excuse my French, but we can, if we use garbage in, we are going to get garbage out, right? So, but we want to get data that's actually meaningful, reliable, and accurate. And with this data, we see that, well, Wilke's study only implemented one subject, and if we match that subject with our height and weight, we get good data. So we're confident about our model. So then we went ahead, looked at all our different static and dynamic conditions on our 11 subjects, and compared marker-based with the IMU-based system. And the first thing we did is we looked at the kinematic output between the marker and IMU. And you see that the blue curve is the marker-based, the IMU is the red. So there's a fairly good match between both systems. We looked at the RMSC, and the RMSC, the average, which is the dotted red line, is about 5, which is what is recommended for modeling, is to be about 5 or under 10. So in average, so we're good on that end. We did also blend Ullmann analysis on these 3 different kinematic motions, and we got no significant differences between our 2 kinematic outputs. So kinematics from antibody from IMU and marker-based systems are good. That tells us that the kinematics from the IMU can be used as input in our system, in our modeling system. So when looking at the contact forces, you can see here in this bar graph, we have all the different static and standing and dynamic positions between the marker-based and IMU-based. We only saw statistical differences between flexion, extension, and walking with weights. You see that there's about 100 newtons difference in flexion extension, and about, I would say, less than 100, about 90 Newton difference in the walking with weights. Now, the question, or that we need to ask ourselves is, well, this is of course statistically different, but clinically, does 90 newtons or 100 newtons make a difference? Now, if you've done experimental work with cadaveric samples or cadaveric vertebra, you're going to see that 90 newtons is not too much. Usually, osteoporotic vertebra, they can range or fail above 1,500 to, let's say, 3,000 or less than 3,000 newtons. So having this difference is not that much, but it tells us that there's a statistically difference between both systems. The other thing is, well, there's a difference if we do longitudinal studies with a single system, let's say this IMU. Well, this gets, we always analyze the same subject with the same system, so the changes are always observed with the same system. So we are happy with the outcomes that we got. We see that the IMU system fairly well matches the kinematics, fairly well matches the contact forces predictions from the L4, L5. So we then went ahead and did additional analysis that's required in modeling when we do this type of process. We looked at confidence intervals, R-square values, and there are very good correlations. The lowest one was in lateral bending, where we got 76%. But overall, the trend is very good to excellent in terms of correlation between the contact forces outputs between the marker-based and IMU-based. Now, the other thing that we need to pay attention when we do modeling is residual forces. So residual forces are going to be present in our model, and we always want to minimize these residual forces. And this, what residual forces refer to, is the forces that are unaccounted for within the model, and these are due to limitations in the structure of the model, the assumptions, or experimental measurements that might be inaccurate. So what are the sources? A little bit of background here. What are the sources of residual forces? Simplifications in the model. MSK models typically approximate the body as a system of rigid segments connected by joints, which may not fully capture soft tissue deformations, ligaments, tendon, etc. Inaccuracies in motion data. Marker-based system can potentially introduce errors. If you have a subject, for example, with a high BMI, if you have them do dynamic motions, well, the skin is going to move, and if the skin moves, the marker will move. But that doesn't mean that the joint, the bony joint, is actually moving, right? You get these errors in the skin, and that might eventually increase the residual forces. Muscle force redundancy and optimization issues. The human body has more muscles than necessary, and there's compensation with different muscles, so these might not be fairly well considered in the modeling process. Joint constraints, for example, assuming a specific joint constraint might not be accurate. And external forces or ground reaction forces that might eventually lead to error. So while we have all of these sources of error in our experimental and modeling process, what we want to do is we want to minimize them. And how we minimize them? By doing a good experimental acquisition of data, by doing a good modeling process, and eventually we can look at what these residual forces are. And we looked at these residual forces, and they are very low, excuse me, in all three axes. They're very well under the 10% or 5% of what's required for a good modeling process. So again, our kinematics are fairly well using IMUs. Our residual forces are good using IMUs. So the other important process here is ground reaction forces. If you remember, the marker-based approach uses a laboratory setting with force plates where we get the ground reaction forces. The IMU uses predictions for these reaction forces because, again, the idea is to go out of the lab and not have subjects come into the lab. So we need a way to either take a force plate out into the outside environment or predict these ground reaction forces. So what we did is we calculated this ground reaction forces and compare both of them. Now, again, to restate what I just said, in the laboratory setting, we use force plates to do the kinematics, and antibody uses the force plate data to perform dynamic analysis. If we use the IMU, we don't have a force plate, so now antibody, what it does is incorporates conditional foot contacts to allow for the GRFs to be estimated. And again, what we did is we wanted to compare how these two results. And when we looked at these two outputs in the lab, we used two plates, one for the left foot, one for the right foot, for different conditions: flexion, extension, lateral bending, walking, etc. And for the IMU system, we needed to match the same experimental conditions. So we looked at two different plates. And if you look at the plates, you see that the forces or the ground reaction forces match very well between the two systems in both plates. When we did Blunt-Ullman in the first plate in all of our subjects, we see that there's not a difference in ground reaction forces. We see that the p-value in walking with weights is significant, but there's this little guy here, this outlier, which we need to further look at that, and you see that it's tends to be the same in all the three different or in all the different motions, but overall very non-significant differences between the predictions and the measured ground reaction forces. We did the same in the other plate, and again, Blunt-Ullman showed no significant differences between the outputs of walking with weight, lateral bending, flexion, extension, axial rotation, and walking. And we were mostly considering or interested in, well, what are the dynamic differences, right? If we just have them stand, we might get better outputs, but we were interested in activities of daily living, so that's why we looked at these ones. So to conclude here, MSK modeling is not a new thing. It's been around for quite some time, but I think that the novelty of this study was that we compared IMUs for estimation of kinematics or for kinematic input into the model, and we looked at the ground reaction forces and the predictions of using this IMU system from antibody to see how we can eventually improve on our workflow process. Now, if you looked at the presentation, we only considered L4, L5, and the rationale for using L4, L5 was that the in vivo data from Wilke looked at the L4, L5. So we validated or contributed to the validation efforts of the model by looking at the L4, L5. But the good thing about antibody now is that it has this thoracic model, and what the thoracic model allows us is, as before, but allows us to get thoracic output for all vertebral bodies. It accounts for thoracic curvature to be modeled. So this is something that we're very interested in because now before, or the model that we use in this presentation, consider the thorax as one rigid body. But the new model, what it allows us to do now is to have the model be flexible in the thorax. So now we can modify each vertebral level based on what we think the curvature at that level will be, and that will eventually induce different loadings in the spine and contribute differently to fracture risk. So this is a very nice thing from Antibody now that we're moving forward and implementing in our research. And like I mentioned, allows for individual vertebral angles to be input. Not only that, we can look at thoracic volume too. So this is an improvement of the previous model that we've been using. And importantly, what I've shown today is just a glimpse of our MSK or fracture prediction process. We can look at MSK modeling, but in our lab, what we're also doing is we're doing QCT-FEA to look at subject-specific fracture properties of vertebra. But if you look at this cycle here, all of this leads to a fracture risk prediction workflow. We can look at modeling, QCT-FEA, kinematics, depending on what your spine curvature is, your muscle weakness, or your muscle condition, you're going to have different kinematics that is implemented in modeling. And there are other factors that affect fracture risk. If you're diabetic, if you have any other bone abnormality, or if you have sarcopenia that alludes to muscle weakness might eventually lead to spine curvature changes. That leads to different kinematics. Different kinematics will lead to different spine loading conditions. So this is a workflow that we are implementing in our lab, with MSK modeling being a key aspect of it to try to improve on the fracture risk prediction approach and overcome the various limitations of DEXA, that it's just a 2D image, which does not account activities of daily living, doesn't account for loading in the thoracic spine, doesn't account for spine curvature, and other risk factors are important for fracture risk. So with that, I'm going to end the webinar with our information about our lab website, our contact information. Thank you all for attending. You've seen the paper that was published that related to this study. I'll be happy to share it if you can access to it, and I'll be happy to answer any questions that you might have now.
Before we go to the Q&A session, I would just like to say a few words about our online resources. So I'll just show my screen again. So if you want to know more about Anybody Technology, then you can go and check out our website, which is called anybodytech.com. And here you can find all of our different events, special dates, and also all of our previous webcasts. You can also go and check out our website called anyscript.org. And this is really our community website for people using Anybody. And here you'll find multiple online resources. It could be our wiki page, several blog posts, and also links to all the repository sites. We also have a forum which is located on this site, and here you can go and ask questions, and hopefully you can get some help from fellow Anybody users, but we also tend to go there and answer questions from time to time. And then I would like to highlight a few upcoming events. So if any of you happens to attend the Gamma Congress in St. Gallen in eastern Switzerland next week, then please stop by our booth there and let us have a talk about musculoskeletal modeling. And also, Aalborg University here in Denmark is hosting a PhD course on advanced musculoskeletal modeling with an emphasis on shoulder, and also this newly developed thoracic spine model, which Hugo also touched upon. So if you're a beginner of musculoskeletal modeling, or if you want to begin with it, then it's a really nice course to start, because it covers really basic modeling stuff, but also more advanced models. And this course will be in April, from April 28th to May 2nd, and it can be attended both on-site and online. So if you're interested in this, then go to our website and there is a link for more information. And then I would like to end this presentation by saying that we maintain a publication list of all of the papers using the Anybody modeling system. And this list has reached more than 1,100 publications, and it's really a great place to go if you want to get some inspiration on how to use the Anybody modeling system, or just see which groups are actually using it. And the list is set up in a way so you can filter it by industry or research area or body part, so you can really be specific to find whatever you are interested in. And with that, I would like to thank you for your attention this far, and